is Heankel Lyons, who's joining us today from the UTSW Medical Center, where she's doing her PhD in the lab of Ben Sabari. She started out getting her bachelor's at Wellesley, and at UTSW, she's been recognized with a number of awards, including the Nominata Award, which is the highest honor that they will bestow on a graduate student. And she was also recently recognized by the University of Utah Biochemistry Department as a Rising Star graduate student, which is a really cool honor, and had a corresponding symposium that she was able to participate in for that as well. And she's going to talk to us today about her work in the Sabari lab, trying to understand the language of transcriptional condensates. So Heankel, go ahead and take it away.
Hey, Jackson. Thank you so much for the intro and for having me in the seminars. So yeah, I'm a grad student in the Sabari lab, and as you probably know, we are interested in understanding how the gene activation machinery is organized in the nucleus. And so I'm sure all of you are familiar with how important it is for these proteins we're studying to get to the right genomic locus at the right time to activate genes. But this is actually quite challenging if you consider how the nucleus is very crowded. And so how could the cell get around this issue? So a hypothesis in the field of biology is that for a chemical reaction with multiple steps and multiple macromolecular complexes like transcription, if you imagine these proteins scatter in the nucleus, and to make the problem worse, there are so many other proteins on the way. If we could concentrate them, this would facilitate a reaction. And so this actually happens in what we call biomolecular condensates. And the disassembly of these condensates can then lead to the diffusion of the proteins, and this can slow down or inhibit a reaction. And so why do we care about this model or hypothesis in transcription? For a very long time, it's been observed that the gene activation machinery is organized in the nucleus. And this has received different names, but again, today I'm looking at this under the lens of condensate biology. So a question is, how are these compartments assembled? So just to get an idea of interactions that are working at these different length scales of organization, you're probably familiar with stable and stoichiometric interactions. And so we've learned this in college, in high school, how protein subunits interact with each other in very specific ways to form complexes. But at a higher length scale, multivalent interactions become relevant to organize these complexes in condensates. And there are different sources of multivalency. Today, I'll be talking about intrinsically disordered regions, or IDRs. And these are quite relevant because many proteins in the transcription machinery are enriched with disorder. And so I'm sure all of you have seen the explosion on papers that connect the phenomenology of condensates and transcription. And it's been quite exciting to see this field grow. But there's also been a lot of skepticism because unlike the traditional model of transcription, where we understand the steps and the proteins and how they interact in very specific ways to activate genes, we don't have that level of molecular rules to explain specificity in condensates. So that's been the central question of my thesis and in the Sabari lab. How is specificity achieved in condensates? So I'll share with you one approach we use to learn about the rules of specificity in condensates. And this is already published, so I'm gonna gloss over the details, but basically we focus on MED1-IDR as a case study. And so MED1, is a subunit of a coactivator. And work from my advisor showed that if you purify the MED1-IDR and you mix it with nuclear extract, it forms condensates. So we were trying to understand what proteins were going into these condensates. And we use a fractionation assay where we can have a brief centrifugation step and collect the condensates in the pellet. So this allowed us to now have the supernatant and the pellet and compare the different enrichment of proteins. And so some of the proteins that were highly enriched in the pellet with MED1-IDR condensates were subunits of the PAB1 complex. So you just learned more about this in the previous talk. And we also had other elongation factors and subunits of RNA pool 2. And to make a long story short, these proteins actually had disordered protein regions that were necessary and sufficient to bring these proteins to the MED1-IDR condensates. And they had this unique pattern of charge. And so you can see these were charge blocks of basic and acidic residues, and they actually match the same pattern that MED1-IDR has. And so lately, we've been trying to use a different approach to get into the rules of specificity. And for this approach, we're using disease genetics of translocational renal cell carcinoma. And so TRCC is a type of kidney cancer where TFE3, a gene, gets fused to a number of partners. And in this case, the DNA binding domain of TFE3 gets fused to these proteins that are now oncofusions, and they drive the disease. And so this was a great case study for us to learn about the rules of condensate specificity in disease, because 2/3 of all cases in TRCC had these recurrent fusion partners, so PRCC, ASPL, and SFPQ. And these proteins don't share any common domains. They don't have any functions in common. What they do share is that they are predicted to form condensates, and they're highly disordered protein regions. And so we wonder if this could be a case study to learn, again, these rules and how they're hijacked in cancer. So one of the first steps was to corroborate that these oncofusions were forming condensates. And so with collaboration with the Bronger lab, I obtained samples, patient-derived xenografts that contain either the wild-type TFE3 or one of the oncofusions that we're studying here. And so I'm showing you immunofluorescence for the wild-type TFE3 or for the oncofusions. And so you can see how the oncofusions have an enhancement in condensate formation. And so we verified this in different ways. And once we were convinced condensate biology was happening, we decided to purify the proteins and mix them with nuclear extract. And so this was the same approach as before, but the difference is that this time we were looking for something commonly enriched in the pellet. And so among all the factors that were enriched in both pellets, we found that subunits of RNA pool 2 were coming up as highly enriched. And so I decided to look into this in cells to verify that this was true. And so I used an engineered cell system from the Spectre lab. And so in this cell line, we have a massive lac array integrated, and I can use this tool to now isolate the protein of interest, in this case, the TFE3 end-term region that gets replaced by the fusion partners. And I can also use the fusion partners fused to the lac array thanks to the lac repressor. So I'm basically creating high local concentrations of these proteins, and this is modeling condensates. And so I can use microscopy to then visualize if other proteins are getting into these condensates. In this case, I'm interested in RNA pool 2, right? So I have in cherry RNA pool 2. And what's great about this system is that it also has a reporter. So I can do RNA fish and detect whether the reporter is activated or not. And so I have more data on other systems, but I like to use this system on my slides just for simplicity. But feel free to ask more in the Q&A if you're curious about other systems. So here, I tested the mCherry RNA pool 2, and I'm showing you how when you have the control or the TFE3 end-term, there is no enrichment of RPB1. But the fusions, they do partition the RNA pool 2. So this was interesting, and so I look into the reporter. And the fusions, again, were sufficient to activate the reporter. And so you're probably wondering what's unique about these fusion partners that the wild-type TFE3 end-term region doesn't have. And so after some analysis, we realized the fusions share an enrichment for this molecular signature I'm showing you here. So they were enriched for some aromatic residues and for some pi-interacting residues, so lysines and glutamines, which can be sometimes partially charged. And they were also depleted for these aliphatic residues. And again, this is all compared to the wild-type protein they replace. And so this helped us rationalize how the RNA pool 2 was probably interacting with these proteins in condensates, because we know the RNA pool 2 CTD disorder forms condensates. It has this consensus sequence, and NMR studies have shown that it can interact with other disorder regions in condensates through pi stacking, which is between aromatic residues, and cation pi interactions. And so it was probably that the tyrosine in the CTD was interacting in these ways with the fusion partners. And so I went back to the Le Carré system, and this time I'm only testing the mCherry RNA pool 2 CTD. And so as you can see here, the fusions, once again, they capture the RNA pool 2 CTD. And when I test a mutant of the CTD where I substitute the tyrosines by alanines, the mutant no longer gets into the condensates. And so this was all, very interesting, but if it's really being used by cancer, we should be able to find it in other oncofusions. So we did an analysis of other oncofusions to see whether they had this molecular signature. So what I'm showing you here is a number of oncofusions that are predicted or have been demonstrated where the fusion partner forms condensates and they are fused to a DNA binding domain. And so what I really want you to get out of this loaded slide is that they have this enrichment for phenylalanines and tyrosines, as well as these pi-interacting residues. And again, they have a relative depletion on these aliphatic residues. And just to show you a few examples I've tested from this set of oncofusions we identified. I tested them mCherry RNA 2 CTD, again in the Le Carré system. And this time I'm showing you the protein region they substitute in the transcription factors, in this case, flea and term. And then I'm showing you the fusion. So in this case, EWSR1. So you can see the fusion, again, is partitioning the RNA 2 CTD. And this is true for FUS, NAP90A, and for a few other oncofusions I tested from glioblastoma and leukemia. And so finally, just to give you an idea of how we tested this molecular signature, I've tested the fusion ASPL. Here I'm showing you as a proof of concept where I substitute the lysines and phenylalanines by alanine. And so in this way, we're swapping the signature, if you will, because alanine is one of those aliphatic residues that is actually depleted in these proteins. So ASPL is a fusion that captures the RNA 2 CTD, but the fusions with these mutations or substitutions capture less of the CTD. And when I test for transcription, they do not activate the reporter. And so it's easy to break something, but can we make gain-of-function mutants? And so to test this idea, I took the TFE3 end term, and I increased the number of phenylalanine lysines. And I did this by substituting those aliphatic residues that are actually depleted in the fusions. And I also introduced some glycines for flexibility in these proteins. So TFE3 end term does not partition the CTD, but these gain-of-function proteins do. And they also activate the reporter. So just a brief summary of the last story I shared. We have used the disease genetics of TRCC to learn how these oncofusions compartmentalize proteins, and we found RNA-pol2 is one of these proteins. And I show you how they use a molecular signature where they have an enrichment for these aromatic and pi interacting residues, and they can interact with the RNA-pol2 CTD. And conferring this signature to other proteins like the wild-type TFE3 can lead to a gain-of-function CTD partitioning. And I like to finish by thanking my lab, my mentor, Ben Savary, who's been incredibly supportive in my grad school, and Prashant, Mikhail, Shubhan, who've been also involved in this project, as well as our collaborators in the Bruger's lab and the Radar lab for the past project on Med1-IDR. And I'd be happy to take any questions.
All right, thanks so much, Heankel. That was really great. I'm curious about when you were comparing like the database of oncofusions. Do you have like an estimate for what what fraction of these oncofusions might be hijacking this condensate formation? And then following up from that, do these condensate fusions, are they strictly DNA binding domains or activation domains, or do you see other like potential chromatin activating domains coming with them?
That's a great question. So I remember from like this large set of oncofusions, the actual percent with this molecular signature is probably close to like 1 or 2. But if you just filter oncofusions with a fusion partner that is predicted to form condensates, then that percent is around 12. And so I think for these filtering, we also use some chromatin binding oncofusions, and so they are actually included. And a fun control we had for this was also analyzing other oncofusions that we don't think should have this signature, such as oncofusions involved in signaling. And so the molecular signature is not found in those other oncofusions that are outside of the nucleus.
Oh, awesome. I think, yeah, Ben was going to jump in with a question too.
Yeah, Michael, that was a fantastic talk and fantastic work. I really enjoyed that. So I was wondering, with this kind of amino acid composition that kind of gets you partitioning into the with Pol2 CTD, how many, like this, it's one way to get a condensate. How many other ways do you think there are? Like when you talk about specificity, are there different classes of things that will drive interactions? And do you have any sense of like how many different ways you could do that?
I feel like that's the question Ben Savary wants to answer for the rest of his life. I mean, I think we're trying to get into that, like what are the flavors, of the different rules of condensates and their function, and potentially how cofactors that are also activating very specific set of genes are contributing to these, and potentially through their IDRs. I think it's still a work in progress. I don't have a good answer other than it's possible there is like enough diversity in these multivalent protein regions to add to that layer of specificity. But I think we're still scratching the surface of this question.
Absolutely. And then another question, if you look at these IDRs that you've been studying from like an evolutionary conservation point of view, I imagine they're not well conserved at any given residue, but do you see conservation of these properties?
Absolutely, yeah. We actually have a manuscript from the lab, and Shubham, he's a computational biologist. So he's actually worked on this question, how are these features, rather than the specific alignment, how are these features conserved across species? and there's actually a very good conservation. So you'll see that manuscript soon. I'm sure he's submitting it anytime soon.
Excellent. Thank you.
Awesome. Well, there you've got a few questions in the Q&A as well. In the interest of time, I'll let you go ahead and answer those in the Q&A box, and I
Sounds
will
good.